going of all too clear. You saw how the food web has changed, and it's making drastic changes to the fisheries in the Great Lakes. We had tribal fisheries last month with the fisheries of the Menominee Nation, and then we had a session on whitefish that Nick presented to us. So we covered the gamut. Now we're going to talk about mass marking lots of fish and the secrets that we learn when we do that. I want to acknowledge our sponsor for this year to help us with our advertisement, Healthy Water Door County. It's been, we're grateful for their support all season long. Tonight, we're going to have Dr. Matt Kornis talking to us. He grew up in Milwaukee, Wisconsin, and spent his formative years exploring the Lake Michigan coast from Milwaukee to Door County. He's earned a bachelor's degree in biology at Lawrence University in Appleton and master's and doctoral degrees in limnology and marine science at UW-Madison. Matt returned to the Great Lakes after a 2-year stint researching Chesapeake Bay fisheries for the Smithsonian, and has been working for the Fish and Wildlife Service mass marking program since 2014 as a researcher and supervisor. And I'm well aware of that because basically I hired him. But there are some handouts out there, so please take them home. And just to highlight one is actually a bill that Congressman Debbie Jingle has submitted to the House for approval, and it is the Great Lakes Mass Marking Program Act of 2025. So if you ever wanted to see what it takes to get a bill passed, this is the bill that they are working on. And it just shows the importance of the mass marking program, that we gain national attention so that it can continue. And I'm going to let him tell all the details of the program, but just it's only 3 pages. It explains, they have to describe the program concisely and so on. And then actually the price tag they have at the bottom is like $5 million a year. So that would be a big boost to the funding. So if you read it and you see the program and you really like it, you may want to call your congressman and say, hey, support Bill 1917. The other thing I want to say is yesterday, I was down in Matt's office, my old office, because there's 3 employees that are retiring. Now, you've heard of the big changes that have been going on with the new administration, and there's lots of incentives to leave. Well, a couple of weeks ago, these people learned these incentives and had to make a decision in a couple of days. And so I think that was last week, and Friday is their last day. So it was quick, but they had a luncheon and I went down. The only reason why I bring this up is that one of the persons was Chuck Bronte, who was on my staff. The mass marking actually started in 2010. So Matt came 4 years in. So Chuck is the one that sort of orchestrated and the architect of putting it together, and was smart enough to find someone like Matt to join the crew back then. Another one was a trailer operator, Jim, and actually he used to work for the company that made these automated trailers, and some of the patents has his name on it. So he was one of the first employees too, because once we saw he was available, because he was actually working for DNR somewhere down in La Crosse. And once he applied and we saw that, well, that's exactly the guy we need, because he helped design these trailers. But because of this change and upheaval or whatever, these people are leaving. And so that means the next in line to lead the Great Lakes Mass Marking Program is Dr. Matt Kornis. So we have just the right guy. And with that, take it away, Matt.
Well, thank you, Mark. Very kind words. Lots of big shoes to fill over at our office, but I love being a part of this program. As Mark said, I've been here since 2014. It's a pleasure to be up here at the Crossroads, up here in Sturgeon Bay. I'm up here quite often visiting my in-laws who are here in the audience tonight. So, no pressure or anything, but so I work with the U.S. Fish and Wildlife Service, and there's a lot of people involved in this particular program. It's a multifaceted program that involves hatchery tagging work, as well as interviews and recoveries from anglers who bring those fish back to shore. And so what I want to kind of do today is give a brief overview
There's an okay up above.
Oh, yeah.
There's something about recording. There you go.
I just signed my life away, didn't I? All right, yes. So what I'm going to talk about tonight, I'm going to briefly touch on the Green Bay field office and give kind of brief highlights about all the work that's going on at your local Fish and Wildlife Service office, and then spend the rest of the time talking about this mass marking program. We do a lot of studies on Pacific salmon and lake trout in the Great Lakes, and I'll be sharing what we've learned on Chinook salmon, lake trout, and steelhead. So a lot of times I get a question asked, well, what's the Fish and Wildlife Service? How are you different from DNR? So DNR is a state agency. Fish and Wildlife Service is a federal agency. Our mission is to conserve, protect, and enhance fish and wildlife and plants and their habitats for the benefit of the American people. That's the nationwide mission. How this our work works towards improving and bringing healthy fish and wildlife populations and habitats, and providing quality outdoor recreation opportunities, hunting, fishing, and other wildlife dependent opportunities. Here in the Great Lakes, our office's main role is to assist state DNRs and tribal agencies with the management of aquatic resources and with invasive species prevention. So I'm going to start by talking a little bit about aquatic invasive species. Our office has 4 main programs, of which mass marking is one. Aquatic invasive species monitoring is perhaps our largest branch. That branch monitors for new invasive species in Lake Michigan and its tributaries. Its focus is on early response and prevention for carps like grass carp, silver carp, big head carp. And they do this through sampling water for eDNA, which gives a signal of what animals are present in the water, and traditional gear, nets and whatnot. You can see a picture of a grass carp here, and then that's a silver carp mount up there. They also work on ruff, gobies, and a few other aquatic invasive species, and coordinate with the states for rapid response should there be a detection. And I will mention there are branches like this for all of the Great Lakes as well. Our office houses the Lake Michigan one. Next, we have a native species branch. Their focus is baseline monitoring for native lake trout and lake whitefish using multi-agency gill nets, running stock assessment models that help with estimating population sizes and harvest regulations. And then more recently, they've gotten involved in acoustic telemetry, which is a cool tool that allows for a fish that has a telemeter tag to provide lots of data points on its location within the lake. And they are out there with a sonar doing some habitat mapping, looking at kind of benthic habitat for fish out there in Green Bay and Lake Michigan. We also house a partnerships and habitat branch. This branch gets away from the lake a little bit. They focus on stream connectivity, culverts and dam removals for tributaries of the Great Lakes. Do a lot of work to get grants and easements for habitat protection. There's some projects on brook trout restoration, native mussels, and risk assessments. And within that, we have the Lake Sturgeon Restoration Program. We have stream-side rearing facilities and release, research on adult sturgeon, including this one that was down at the De Pere Dam. They should be back in maybe a week or two. And then we also help operate a fish lift that moves spawning lake sturgeon upstream in the Menominee River. So that's a quick crash course in all the programs at the Green Bay field office, except for our branch, which is the Great Lakes Fish Tagging and Recovery Branch. So to get into some details on the work that we do, it starts in the hatcheries. We have a photo up here of one of our auto fish marking trailers. These allow us to mark and tag fish at a rate of, on average, about 8,000 an hour, which amounts to about 2 to 3 fish every second coming out with an adipose fin clip to indicate that they're from the hatchery, and a coded wire tag that gives us data on where they were stocked, when they were stocked, how old they are, and what their genetic strain may be. And that allows us to answer a lot of interesting management questions about return on investment for these fish, which locations are producing the best, which genetic strains, how much movement's happening, from fish from point A to point B. So I'll go through kind of all these in detail, but just in a general sense, our program is a multi-agency collaboration that is among federal, state, and tribal agencies. It's coordinated out of our Fish and Wildlife Office in Green Bay, but it involves all 4 of the Departments of Natural Resources on Lake Michigan, Wisconsin, Michigan, Indiana, and Illinois, as well as multiple tribal nations. Began in 2010 to address questions on the survival and contribution of hatchery fish, natural reproduction, movement, growth, and other measures for salmonines. While the states have management authority, Fish and Wildlife Service can help coordinate across jurisdictions. So that's a bit of the collaboratory role that we have with our partners. Presently, this is entirely funded by the Great Lakes Restoration Initiative. So these are non-permanent funds that are supplied to us through the Environmental Protection Agency. We've received lots of support from them over the last 12 years, and I'm very appreciative of that. But we've also received a lot of support from you and from other anglers and members of the public. All of the data we collect come from sport fishers. And you know, it's all voluntary, whether people interact with our techs or not. And you know, it's had enough support so that over the last 12 years, we have data on over 172,000 fish landed by open water anglers in Lake Michigan and the United States waters of Lake Huron. So that gives us a pretty substantial data set. It's been lots of fun for me to play with. So tagging and marking a hatchery fish. So I already talked a little bit about this. We have 4 main prongs in our program, the first of which is the tagging and marking of these fish. We also have data collection and tag recovery from the sport fishery. I touched on that a little bit. This is where we collect biodata, length, weight, sex, maturity, the fin clip, whether or not there's a tag, and a bunch of other data to help us answer some of these management questions. In the fall and the winter, we have tag extraction, which, unlike the actual tagging process, is not automated. We have to pull each tag out by hand. It's a little bit painstaking, but it's important to get those data back. We also age wild fish to give us some estimates of what's going on with the wild counterparts, these hatchery fish. And then we do things like what I'm doing right now, which is data analysis and support for management and outreach. And yeah, I have not aged terribly well. So marking and tagging, what you're looking at in this figure is the number of fish that we have marked every year since the program's inception in 2010. And you can see that with the exception of 2020, for the reason everyone would think, we mark between 9 and 10 million fish every year. So last year we marked a little over 3 million lake trout, 3.5 million Chinook salmon, and close to 3 million steelhead, as well as a handful of brook trout that are actually stocked in Lake Superior as the coastal brook trout rehabilitation. You can see in this figure, we have lake trout in green, Chinook salmon in blue, and steelhead in pink. So you can kind of see those numbers have been pretty consistent since 2018. We do have a change coming to this year. So at the request of the states, we have diverted the funds that were used for steelhead. We've had 7-year classes of steelhead tagging, and instead those funds are going to be used to tag coho salmon to try to learn about that species. And so we are actually going to wrap up our first year of coho tagging this June, and have already tagged many of these fish. So this is a fun part here. A lot of people wonder how this tagging works. So would you guys like to see how the trailer works? All right. So I'm going to show you 2 videos. So this first one, we're going to see fish that are in a holding pen. This right here is a big holding pen. And one of the main concepts behind the design here is these fish like to swim upstream. So we provide current that will encourage the fish to swim in the direction we want them to go, which is against the current. And so what you're going to see here in this video, these are fish that are swimming up that pipe into a big sorter up here. So this holding pen is going to go down a length sorter. You're going to see these big red lights, and those lights are very rapidly measuring the length of these animals as they fly through here. Getting quick length estimates because these tagging lines are calibrated for very specific size ranges of fish. All that clicking is you're hearing them being diverted to these 6 different tagging lines. My kids love this video. We talk about how the fish go down the water slide. They go into these little holding pens here, and then there's current actually going upstream, so they will swim into these tagging chambers, and you're going to see this brown thing come down. That's clipping off an adipose fin. You can see a little fish right in the middle there if you have good eyes. And at the same time, they're receiving a coated wire tag from one of these blue tag injectors. Those tags go right in the snout. These coated wire tags, they're a very tiny metal filing. I think it's like 1/64 of an inch, something like that. It's 1 millimeter, whatever that comes out to in English. And they're etched onto those tags is a batch code that all the fish stocked at the same location, say 100,000 fish stocked here in Sturgeon Bay at Strawberry Creek, they'll all have the same coded wire tag telling us those fish were stocked at Strawberry Creek in 2025 and came from the Lake Michigan Chinook genetic strain. So the other part of this is what happens outside of the hatchery. Oh, that's loud. Hold on a sec here. That was way too loud. Whoops. It's not going to let me, so we'll reduce the volume on my computer. How about that? That's better. All right. So this is up at, actually, I don't remember which hatchery this is, but you can see all the raceways with all the fish. They're organized into different raceways so we can keep tag lots separate. All the hatchery staff help tremendously with that and have that all set up for us when we arrive. This is a big water pump. So this is pumping in all of the water that goes into the trailer, coming in this big hose. So that comes in the front. And then what you're going to see as we walk towards the back is the fish that are clipped and tagged go out the back and get piped into whatever raceway is ready for them. So you'll see kind of this piping back here. And then we do a quality control check. So every hour or two, we sample at least 100 fish from the back here. You'll see some fish coming through. And we investigate them for the clips and the tags to make sure we're getting accurate clips and tags. About 99.5% come out with a fin clip and about 96% with the tag. So we have pretty good success rates. There are a variety of quality devices inside the trailer. If imaging suggests we missed a fin clip, it'll be kicked out to a reject bucket. There's also a magnetic sensor that'll let us know if a fish has been tagged or not. And if it's not tagged, that gets kicked out as well. And then those are handled by hand. So that's part of how we get such a high success rate there in the hatcheries. But all that tagging and marking in the hatcheries doesn't mean too much if you don't get the data on the other end. So that's the other major part of our project is the tag and data recovery. So what you see here on this map is all the ports that we sample in a given year. Those are the red dots, and that's where we're interacting with sport fishers and collecting data from their catch. And if those fish are tagged, we collect the tags at that point. These black stars are where we station our crews. And so this is one of the most extensive sport fishery surveys to collect these data. About 15,000 fish a year on average. Last year was a little above 13,000 fish that our teams put our hands on. But we're surveying, I think those numbers are a little out of date. It's between 40 and 45 ports every year. The baseline surveys, the nets, those are set on the bottom. We get a lot of lake trout in those, but not so much with the Chinook salmon, the steelhead. Our sports fish survey is the most abundant source of samples for those Pacific salmon, and that's allowed us to interact with a lot of other research institutes around the Great Lakes to do cool things like look at diets and other things that I'll try to get into. We also have our lab operations. As I mentioned, these tags are extracted by hand. You can see a picture of the etched code on the right there. So that particular code corresponds to some lake trout that were stocked in the middle of Lake Michigan in 2010, and you know, it gives us that information. We've recovered close to 160,000 tags so far, not only from our survey, but also from state creel surveys, weir surveys, tribal commercial fishing, and voluntary angler returns. And then aging wild fish, this is something that's important because the age of the fish plays a strong role in estimating population size and survival. And for those who aren't aware, we can look at calcified structures like scales or otoliths, and it's sort of like rings on a tree. Now, I will say I cherry-picked this photo. It looks great. Most of the samples are a little harder to interpret than that, but that's the main concept. So that's another major aspect of our program. And then on the back end, in terms of what we use these data for, we've helped answer questions about wild reproduction of these fish, movement patterns, survival and growth rates. These data are regularly used in the management and management models that are used to inform policy decisions on stocking and harvest. And then this field survey, because we put our hands on so many fish, we've been able to collaborate on things like looking at sea lamprey wounding. This was a study where we found that Chinook salmon are also an alternate host for sea lamprey, as well as lake trout, which is the primary host. Going on almost a decade of diet work, looking at the diet of these animals in both Lakes Michigan and Huron. where we basically found that Chinook salmon and coho salmon are kind of obligate feeders. They eat almost universally alewife, regardless of how abundant alewife are in a given year. Whereas steelhead, brown trout, and lake trout, they also love to eat alewife. But if there's a low alewife year, they are a little more resilient and start feeding on things like gobies or invertebrates or whatever they can find. We're involved in a couple of collaborations with Central Michigan University. We collect these ear bones of the fish, and the calcified structure of those ear bones. They actually bond to the trace elements in the streams. And so that then gets locked into place at the time at which those fish are hatched. Literally, the way this works is you shoot a laser at it and it spits off these chemicals, and you can then match the trace elements to that in the environment to estimate basically the location where these wild fish came from. So that's a really cool thing we're doing with steelhead, Chinook salmon, and coho salmon. We've looked at the genetic composition of wild lake trout, figuring out which strains are contributing to recruitment, and then PFAS, biocontamination, which in Lake Michigan wasn't as bad as we had feared, is kind of the take home from that. So that's kind of a brief overview of all the things that we look at. And now I want to kind of get a little more specific on the three main species that we've studied. So I'm starting with Chinook salmon. One of the things we moved really early on is that Chinook salmon move all over the lake for most of the year. So what you're looking at in these figures, if we start on this map here, every fish in this map was landed in Manitowoc. So that's what you see, that gold star there. And the red circles correspond to the amount of fish from different stocking locations around the lake that were caught in Manitowoc. So if you're an angler and you go catch your Chinook salmon in Manitowoc, it could literally have come from anywhere. And what we see is that this pattern is very similar regardless of port. So I put Frankfurt on the other side here. This port's opposite end of the lake, from Manitowoc, and yet we see a very similar pattern with where those fish come from. So this isn't a huge surprise to us because, Chinook salmon are native to the Pacific Ocean. Great Lakes are big, but they're no Pacific Ocean. So it stands to reason what we think they're doing is they're following around alewife and moving around in search of prey for most of the year. So how this affects local fisheries, this is a series of pie charts I put together for a talk a month ago. What we're looking at here are tagged Chinook salmon that we landed between Algoma and Manitowoc, nearby here in Wisconsin. And we're looking at a pie chart of their stocking locations. So this pie chart here shows fish stocked, fish that are caught in this area from April through July. And so the black part of the pie chart, that's fish that were stocked in the same place where they were caught. It's Algoma, Kewaunee, Manitowoc, Two Rivers area. And you can see that represents 10 to 15% of that pie. Elsewhere in Wisconsin was actually a fairly substantial chunk, and then part of that's because we have really high survival of Chinook salmon stocked in Wisconsin. But you can see that fish that were landed here in this part of the lake came from all over. And we can see that persisting into August. But we did learn pretty quick that even though these fish, after they're stocked, only hang around their stocking locations for 2 to 4 weeks, it's during that critical stage when they imprint on the location. So we do see evidence of natal homing. This is a figure from September and October. You can see that that black pie chunk from fish that were stocked in this area is now close to 3/4 of the fish that were landed. So even though stocking location doesn't really matter for catch location for Chinook for most of the year, during the fall, the stocking location really does matter in terms of supporting those fall stream fisheries. Another thing we learned with the Chinook is that the Chinook stocked in northern Lake Huron pretty much universally come into Lake Michigan to feed. So this map on the left shows Chinook salmon that were stocked in northern Lake Huron, and each red circle is proportional in size to how many were recovered in a given area. So you can see it's actually around here near Sturgeon Bay, Green Bay, and northern Lake Michigan where we see most of those Lake Huron fish, but they are pretty much all over. And the main reason for that is that I mentioned Chinook salmon's main prey is alewife. Well, the alewife population in Lake Huron collapsed in 2004, 2005 for a variety of reasons, including predation, but also harsh winters and invasive species and a number of things. And as a result of that, we participated in a study with Michigan DNR that kind of compared these data with data from before the alewife collapse. And this is definitely a change. Those Huron fish used to stay in Huron. Now they're coming over to Lake Michigan to feed.
over 94% in any given year. On the right, though, we see those same fish stocked in northern Lake Huron during what would be considered the spawning season. And you can see now that those fish do return to that stocking area in northern Lake Huron. Another interesting thing is we don't see any reciprocal movement. So the stat I have here is 0.2% of Lake Michigan salmon. I think we've recovered maybe 24 Chinook salmon stocked in Lake Michigan in Lake Huron over the course of the last 10 years. So it's very much one-way movement. Another thing we do every year is we estimate the wild recruitment of Chinook salmon. This gives us a solid estimate on their population size, which helps the states manage for predator-prey balance, which is the goal to sustain the fisheries to make sure that we have an appropriate amount of predators for the available forage base. So this is based mostly on the fin clips. A hatchery fish is going to have that adipose fin removed. That's the top. And the wild fish, we see that that adipose fin intact. We look at wild production at age 2. That's the highest confidence we have because we see the most fish at that age. At age 1, they're still a little small, and wild fish tend to be a little smaller than hatchery fish. So age 2 gives us the least biased estimate of production. And basically, we use the percent of wild observations with the known number of stocked fish in a ratio to estimate how many wild fish must have been there to produce that observed percentage. So what we see for our percent wild is it's been pretty consistently between 60 and 70% wild for Chinook salmon. They've really adapted to life in the Great Lakes. Again, these are not native to the Great Lakes, but nonetheless have found a way to have really substantial wild production, with the exception of 2013, which I'll get to in a moment. If we now combine those percentage data with the known number of stocked smolts, we arrive at this figure, which is on a handout back there. So the y-axis here is the number of smolts in millions by year class. So the x-axis here is the year class of the fish. The stocked smolts are in orange. That number is known. The blue is our estimates of wild production. And so a couple of things to point out on this figure. We didn't have a field season in 2020, so that's a slightly different shade because that's actually the average value. The wild estimates the average value of 2018, 19, and 21. And then 2023 is a different shade because that year class, we only have data on age 1 fish from last year. Those fish will be age 2 this year, and we'll be able to update that number with a more formal estimate. But in terms of our numbers for the last couple of year classes, you can see a general increasing trend in wild production. Estimate about 7 million from the 2022 year class and about 9 million from the 23 year class. But there is a lot of variability here in the wild production. So if we take those stocked fish off, I kind of see 3 major periods of time in this figure. So if we look from 2006 to 2012, we're seeing wild production of about 6 to 8 million wild fish a year. And then we hit 2013. So what happened in 2013 is there was a historic drought during the fall. Historic low numbers of fish showing back up to spawn at Strawberry Creek and other areas. About half the lake was affected by this. And then that was followed by the 2013-2014 winter, which is when there was like a polar vortex just sitting over the lake for 40, 50 days. I remember this because that was the spring I moved back from Maryland, and we arrived here in mid-May and it looked like February, and we're like, we've only been gone for 2 years, but we forgot that it's not spring in May. But that was a particularly delayed spring, and that could have affected the the matching up with these smolts trying to get food sources after they were stocked in the lake, or actually after they smolted out these wild fish. So after that 2013, we had a period of time where we had lower production, and I'm speculating that's in part because we had lower wild fish from that 2013 year class. It took some time to build up that spawning stock again. And then the last couple of years, we've kind of been back to the amount of production we saw at the first part of this time series. So one thing I want to mention about wild fish. So I did show you these pie charts earlier. These were just stocked fish. These were just fish that were caught between Algoma and Two Rivers. These are just hatchery fish. If we add the wild fish to these pie charts, you can actually see how important the wild Chinook salmon are to the local fisheries here. So April through August, we're seeing that number, close to 2/3 of the landings are wild Chinook salmon. And then we do see still that importance of those locally stocked fish in the fall. One thing we looked at, kind of this figure is statewide for Wisconsin. we're looking at kind of just general patterns here for Chinook salmon. So you're seeing the percent of fish captured by month. And the lines to pay attention to are the red line. Those are fish that were stocked in the same area where they were caught. And that's less than 10% from June through August. And then the gray line is the wild fish. Here in Wisconsin, about 2/3, but then you can see that comes down very much so during the spawning season. And that led us to believe that those fish are likely not spawning in Wisconsin. When we look at the same figure for Michigan, that gray wild line goes up. And so one thing we did is we looked at the fall catch rate of wild Chinook salmon, which we have in this heat map here. So this is the observations of wild Chinook salmon per sampling day in that fall period. And so this kind of confirms that northeastern Lake Michigan is likely a focal point of wild production. We're assuming these wild fish are going back to the same natal areas where they were hatched, and so that these catch rates likely reflect, the spawning densities and the amount of production. So that's pretty cool. And that's another one of the things we've learned about Chinook salmon. I start with Chinook because the Chinook study was from the 2011 through 16 year classes. And then we continued to clip those fish. So we have the most data on Chinook salmon. When we look at those 2011 through 2016 year classes, we're actually able to estimate survival by looking at the return rates relative to how many fish were stocked. And we interpret that as a relative survival. And what we see is that these warmer colors in the map indicate higher survival. So in the Wisconsin waters, for whatever reason, we see a really good survival rate of juvenile Chinook salmon. Some of the reasons we think this may be, we have the prevailing jet stream moving west to east. That pushes the warm water into Michigan. We had colder water in the shoreline here, and that's a better temperature for these Chinook. We also have a lot more rocky substrate on the Wisconsin shoreline than in the Michigan shoreline, for that same reason that those prevailing winds will push the softer sediments, the sands and whatnot eastward. And those rocks support a rich array of invertebrates, little bugs and whatnot that smolts for Chinook do rely on before they get big enough to fully feed on fish. Some other things we've noticed is that we had really low survival for Chinook salmon stocked in Green Bay and in MM6, which is the unit by the Little Manistee River. And so what we think is going on in Green Bay, compared to the rest of Lake Michigan, it's warm and it's got a lot of predators, walleye, cormorants. You folks likely know all this. So that's likely keeping survival of Chinook salmon stocked there down. And then in MM6, there's a pretty unique situation. The Little Manistee River, the Michigan's main egg take facility is there. And it is, I want to say 9-ish miles upstream of the coast. So there's a lot of stocked fish. When they stock in that particular unit, they're stocked pretty far upstream. They have to go through that stream. They have to go through Manistee Lake, which is one of these drowned river mouths. And there's likely a lot of predation going on as they try to get back out to the lake as well. So that is, these are all hypotheses. We don't know for a fact, but these are things that we think might be going on because it would make sense if that's the case. We also saw a decrease in relative survival of stocked fish from 2011 to 2016, and the reasons for that are less clear, but we are starting to tag these Chinook again this year, so we'll be able to see and check in how things are doing. So next, I'll move on to lake trout. So lake trout are a native fish to Lake Michigan, the native apex predator. I presume Marcoli's favorite fish, maybe. He spent a lot of time working on lake trout, at least I know that. And so what we do with our mass marking data is we look at the relative abundance of fish, and that's calculated as fish per sampling day. So we have technicians out there examining fish that are brought into port by the anglers. And so for lake trout, we kind of summarize this for 3 large regions, Southern Lake Michigan, which will be in dark blue, Northern Lake Michigan, which will be in light blue, and then Lake Huron. And I should say there's a lot of data on this as well from the fishery independence surveys, the bottom set gill nets, and by and large, the data are more similar than they are different. We won't get into the nuances, but we do see these signals in multiple sources. I'll kind of just skip over those methods. But basically, we're only recording a data point when we feel like we have enough fish to look at this. So this figure is the relative abundance of wild lake trout based on those observations per day. So every fish in this figure is wild, and we're looking at those three main regions and the trend lines for Lake Huron in orange, Southern Lake Michigan in dark blue, and Northern Lake Michigan in light blue. So we can see that the frequency with which we've observed wild fish has gone up substantially in Lake Huron over this time series, and up a modest amount in both Southern and Northern Lake Michigan in this time series. So in Southern Lake Michigan, we started seeing this increasing pattern as early as 2014. For Northern Lake Michigan, we didn't really see, it was pretty close to zero until right around 2020. We kind of had a blip year. There's a lot of natural variability here in our observations. So you kind of take a step back and look at the trends, but we do see positive movement for wild recruitment for lake trout. And one of the reasons for this may likely be the fact that alewife are in relatively low abundances here. Alewife, even though lake trout love to eat them, they feed on larval lake trout. And if you eat too many alewife, you have a thymidase deficiency, a critical enzyme for processing thiamine that can result in an early mortality rate. So there's definitely a correlation there with what we have seen. Now, next I want to look at comparing the wild and the hatchery lake trout for each of these sections. So this is Lake Huron. In all of these figures, you're going to see hatchery lake trout in orange, wild in gray, and the total in black. And again, we're looking at an estimate of relative abundance based on our observations. So for Lake Huron, what we see is that the hatchery lake trout catch rates have been relatively stable, as stocking rates have been relatively stable, and that the total catch is reflecting what we see with the wild lake trout in Lake Huron at this point. If we move into Northern Lake Michigan, we see our data for hatchery lake trout bounce around a lot, and that's in part because in certain years we didn't have enough samples to include all of the districts. So that's some of the variability you're seeing there, but the main take-home point here is while we've seen some modest improvements in the amount of wild lake trout, the total catch rates really strongly reflect the hatchery trout. And for a variety of reasons in the northern part of the lake trout tend to be harvested a little earlier. There are commercial fisheries up there. There's also more sea lamprey up there. And as a result of that, there's generally less wild production in Northern Lake Michigan at this point for lake trout. In Southern Lake Michigan, we have a very interesting pattern. So this is where we see that wild lake trout, pretty modest improvements, nice improvements there. But hatchery lake trout have come down a lot. And that's because of stocking reductions in this part of the lake over the last 2 decades. So while early on in our time series, we saw that the total lake trout rates were very mirroring the hatchery, at this point, they're mirroring kind of a little bit of both. And one thing I want to point out, Mark was actually talking to me about this at dinner. We used to talk about the ratio, the percentage of wild lake trout, and use that as a measure of whether or not we were seeing lake trout reproduction in the lake. And that's no longer a good measure for this. And this figure shows why. So we have our catch rates on the left axis, wild lake trout in gray, hatchery in orange. And of course, the percentage is going to be affected by both the orange and gray lines. So on the right, we have the percent wild lake trout. And you can see this pink line, the percentage of wild lake trout has climbed a lot. We're more than 50% wild in the southern part of Lake Michigan now. Which sounds great, but you can see that that's driven not only by increases in wild lake trout abundance, but also decreases in hatchery. And I did say this is kind of related to stocking rates. This is a figure showing basically the amount of lake trout stocked in this part of Lake Michigan over time. And lake trout in the sport fishery, we usually catch them at ages 5, 6, 7, 8, 9, 10, and older, but primarily ages 6 through 10. So if we put our hatchery catch rate on this figure and we subtract 6 years, this is what it looks like. And you can see that decline in the hatchery catch rate more or less mirrors the decline in stocking from the 2012 to 2018 year classes. There was a more recent stocking event that will likely kind of continue to see this type of pattern. So another thing we look at for lake trout is their movement. So unlike Chinook salmon, which move everywhere, lake trout, their ranges are a lot more limited. And so I've got 2 figures here. The one on the left is a pretty clear example. So all the fish in this map on the left were stocked in WIIM, which is highlighted in pink on this. It's called that because it's got a part of Wisconsin, Indiana, Illinois, and Michigan. So WIIM. we're very creative sometimes. So what we can see here is that those black circles, those are the catch rates proportional in size, and you can see most of the lake trout that are stocked in WIIM are caught in WIIM. The figure on the right is a little more complicated, but it gives us a very general sense of movement in Lake Michigan. So what I did on this figure is I took the 3 locations that are offshore, where we tend to see the most amount of lake trout movement. And I've done it's called a cumulative proportion plot. So at 0, we have 0 of the lake trout that were caught. And eventually you can see they all plateau at 100%. So that y-axis basically shows at what distance we catch however many number of fish. And the key take home here is this line, 100 kilometers, is about 62 miles. It intersects with the fish that were stocked in Illinois in yellow, the southern refuge in blue, and the northern refuge in purple. That 100-kilometer mark intersects at about 72 to 82% of the recoveries. So take home here is about 3/4 of lake trout are caught within 60 miles of where they're stocked, which is very different from what we see with Chinook salmon. We do know with the stocking plan and the management plan for Lake Michigan, there are priority stocking zones. 2 of the big ones in Lake Michigan are the northern refuge and the southern refuge. Julian's Reef in Illinois is another priority zone. And one thing we've learned is that these refuges, even though they're offshore, are source populations for nearshore fisheries. And we see that from this map. So in this map, we see in red, those are circles representing the lake trout stocked in the southern refuge. And blue circles are stocked in the northern refuge. And you can see lake-wide between these 2 refuges, we're seeing a substantial amount of lake trout caught in these nearshore areas. We do see big circles within each refuge, however. So we also have learned that those refuges are providing some protections as well, because a portion of those lake trout appear to be staying there. And we're going to learn a little more on the details of this from those acoustic telemetry studies that I mentioned way back at the beginning. We also know from our data sources that lake trout stocked offshore have had higher survival rates, which makes sense. These are historically important spawning grounds for lake trout, but they're also, offshore. They're away from fishing activities. It's illegal to fish within the boundaries of the refuge, and there's good solid habitat out there. So we actually see for the blue tones are fish stocked offshore. These warmer tones are fish stocked near shore. The letter codes represent statistical significance. So if we're going to speak very generally, fish stocked at Julian's Reef, which is here in Illinois, and the southern refuge do have substantially higher survival rates than other locations. Northern refuge and these nearshore zones were very similar, and then for whatever reason, lake trout stocked in nearshore west. Those lake trout never really survived very well, and at this point, no more lake trout are stocked in this area. Basically based on constituent demand and management decisions to focus on those offshore stocking sites. So that's what we have for lake trout. And then last but not least, I want to talk about what we learned for steelhead. So steelhead's the most recent fish that we've started working with. Although I say most recent, we have been tagging them for 7 years. And what we've seen so far is that the overall percentage wild fish in Lake Michigan, it has ranged between 32 and 50% based on each year class. And, in science, it's nice to see similar numbers from different approaches. So I mentioned the microchemistry studies with the lasers shooting the ear bones. And so from that study, they actually estimated about 40% of their observations were wild. So 2 completely different approaches, tagging and microchemistry, coming up with very similar numbers. So that gives us a lot of confidence in the amount of wild steelhead that are out there. So we do a similar analysis to what we do with Chinook. We know how many steelhead were stocked, that's in orange. We know the percent wild from our observations. We can do a ratio to figure out how many wild fish must have been there. I do want to note right away in 2020, that's the year class, there was no egg take in Michigan. This is something I actually called Michigan about when I made this figure. I wanted to confirm that wasn't an error in the data. And they said, no, we didn't have egg take because of COVID. So you can see these orange stock bars, that decreased in 2020. 2020 is a bit of a weird year because of that. So I have it in a little bit of a different shade. But otherwise, steelhead stocking has been really consistent. Wild production has been more consistent for steelhead than it has been for Chinook so far, but we do see of these first 3-year classes, a little bit of a higher wild production than the more recent year classes. We're not quite sure why, but we're going to continue to get data on these fish for at least the next 4 years. Again, the last fish were tagged now, but we tend to see these fish at ages 2 through 4. So we're going to continue to collect data on these fish for years to come. So one of the things we're curious about their movement rates. Are they going to be more like Chinook or more like lake trout? And they are in the same genus as Chinook. So it didn't shock us to learn that they are very similar to Chinook in the movement patterns. In that they're moving lake wide. So this example map here, we see steelhead that are stocked in Indiana, and those purple circles are proportional to the catch. So all those fish were stocked in Indiana. You can see they're being caught lake wide. But we see less movement between the lakes. So you remember most of those Chinook salmon from Lake Huron were coming over to Lake Michigan. For steelhead, it's only about 25%. And we think this is because the steelhead are more adaptable in terms of their diets. And can find more food in Lake Huron and therefore aren't as motivated to move great distances. If we want to look specifically at fish that are stocked in Wisconsin, this figure on the left shows steelhead stocked in the unit we call WM4. These are our kind of standardized management units. WM4 is that Algoma, Manitowoc, Two Rivers, and Kewaunee. And we can see that those steelhead are being caught all over the place. Now, Wisconsin also has a tag lot that it's used for all of its small tributaries, things like Hines Creek, Hibbert's Creek. Nick could speak more on that for specifics, but basically we don't know exactly where these fish came from, but they're being caught all over the place as well. And I'm highlighting those because they're going to be important later when we talk about survival. And then fish that are stocked in southern Wisconsin, we can see they're moving all over the place as well. So who's catching who's steelhead? And this has been a message we've given to the states, that everybody's catching everybody's steelhead. So all the states are relying on all the states for their catch rates. If we look at these pie charts, we're looking at fish that were stocked in Illinois are in green, fish stocked in Michigan are in blue, fish stocked in Indiana are in purple, fish stocked in Lake Huron are in orange. That light purple is basically Indiana, but there's some fish stocked in southern Michigan, and then gray is Wisconsin. And so caught in Illinois, Indiana, Michigan, Wisconsin, we can see those pie charts are very similar, with the exception of Indiana fish really doing well in Indiana. And so, you know, if we look to explain these patterns, it's easy enough to do, because if I show you this pie chart, this is the amount of stocking in each of the states. And so if we compare those pie charts, we see Indiana steelhead are a little bit disproportionately high. So that would lead us to believe that they're surviving better. And we do confirm that later with more rigorous analysis. But otherwise, we're basically seeing catch rates that are very similar to stocking rates for steelhead based on their location. And again, here, if we focus in on that area from Algoma down to Manitowoc, we can see that the fish that are caught here, some of them are stocked nearby, that black bar again. But again, that's about 10%. And the rest of those steelhead, for stock steelhead, are coming from elsewhere. And again, we can add in the wild fish. We do see about 40% wild for the recovery rates here in that area of Wisconsin. So that gives you a kind of a total picture of the fishery, wild versus stocked, and along with stocked fish coming from all the different states. So to look at survival, I can get briefly technical here. We have survival rates for these fish returning at ages 2 through 4. We actually see them up to age 10, but the most samples we get are from 2 to 4. So that's what we look at. But the problem is we didn't have a field season because of COVID in 2020. And as a result of that, each of the year classes have different ages at recovery. So to kind of get across that challenge, because that has the potential to bias the analyses, we did something called standardization, where we basically are expressing these survival rates in the units of standard deviations from the mean, from the average. And so on these coming figures, you're going to see a value of 0. That's going to be average for that year class. Positive number means better than average. Negative number is worse than average. And 1 and -1 are equal to plus or minus a standard deviation of the mean for folks who are, you know, want to nerd out on stats with me. So this basically removes the bias from different ages being used in different year classes. So the first thing we looked at, so for steelhead, they're actually stocked at 2 different stages. We have steelhead that are stocked as yearlings, you know, bigger fish that have been alive in the hatchery for about a year. And then fingerlings, fish that are stocked, you know, in the fall, in the same year they were hatched. A lot of the stocking decisions there are based on hatchery logistics. A lot of those fingerlings are stocked as fingerlings because they need to get them out of the raceways to make room for whatever species they're rearing next. So as a result, we don't see a ton of potential to change how these fish are stocked. But what we have learned is that the yearling fish have about 2.5 times higher survival rates than the fingerlings. So here is that figure showing relative survival. Again, that's standardized units, so 0 is your average. You see the fingerlings typically have below average survival. The yearlings are typically above average. And another way of looking at this, the fingerlings represent 23% of the steelhead stocked in Lake Michigan, but only about 7% of the sport harvest. We can look at this another way. If we look at fingerlings and yearlings that were stocked in the same areas of the lake, there's 3 such areas in the lake: Indiana, basically central Michigan waters, and southern Michigan waters. And in this case, blue is fingerling, orange is yearling. We can see those fish stocked in Indiana, even the fingerlings are not doing bad. Again, 0 is an average value, and we see a value above average for fingerlings on average. But what we see for the other 2 locations is negative for the blue. And in all 3 of these locations, we see that the yearlings are outperforming those fingerlings when we control for the stocking location. So we're pretty confident of this life stage effect. And so for the rest of these analyses, we're really going to focus in on yearlings. And so this figure is looking at the survival of steelhead yearlings based on their stocking location. And I'm not going to spend too much time on this figure because it's much easier to look at this on a map. But I basically want to note that we have 3 districts that performed really well, and those are going to be color-coded blue in the next figure. A whole bunch of stuff that was around the average, that's going to be color-coded yellow, and then a couple districts that were below average, those will be color-coded red. And again, average here is that dashed red line. So when we put these on a map, like I said, much easier to understand now. This figure on that we're looking at over here is our relative survival rates. And so those blue units are the areas where we see pretty decent survival. And in this case, the pattern is a little different from what we saw from Chinook salmon. Generally speaking, we see pretty good survival for steelhead in the southern basin of Lake Michigan. And I'll say Illinois is not on here because they stock only fingerlings. This is a yearling only map. And then we have decent survival in the north, but our sample sizes are lower up there. So we don't have as much of a confidence in that. And then there's this blue box, Wisconsin small tributaries. I said that'd be important later. That actually is one of the highest surviving tag lots in Lake Michigan. For whatever reason, those fish that are spread out throughout all the small tributaries here in Wisconsin have really good survival rates. And they're represented as a box because they're really spanning the whole Wisconsin coastline. If we look at our low survival areas, we do see a commonality with Chinook salmon. The Green Bay, we don't see a very good survival rate of steelhead stock there. And then for whatever reason, actually right here in the Algoma to Manitowoc region, we see relatively low survival of steelhead plants. And we're not sure why that is either. Initially, we thought it might have to do with the differences in size at Wisconsin hatcheries versus other hatcheries. But that actually didn't bear out after we had more data. So, you know, that's a little bit of a mystery, but, you know, what's interesting here is that we do see some areas of all these states producing a good amount of steelhead. Wisconsin small tribs, Indiana does really well, and then southern Michigan. And then just for funsies, this figure here shows the stocking locations. This is available on the web. It's called the Great Lakes Fish Stocking Database. So if you Google that, you'll get to this interactive map where you can pull up different fish and look at where they were stocked. And so in this case, these circles are proportional in size to steelhead stocking. So you can see where the fish are stocked and their relative survival rates. Next for steelhead, there's a lot of different genetic strains stocked in Lake Michigan. Skamanias tend to have a higher survival rate. I'll preface this by saying we have yet to see any statistically significant results. Basically, for it to be statistically significant, we want these p-values to be less than 0.05, but you can see we're getting kind of close in some of these. So I can talk about this as a trend. Here we're controlling for location again, and we can see that in Indiana and NM8, Skamanias did a fairly bit better than Michigan winter, but we don't really see much differences among the other genetic strains. Wisconsin stocks Chambers Creek and Ganaraska, as well as Skamanias and Arlee. These are 4 areas of Wisconsin where we have Chambers and Ganaraska stocked together. And when we control for location, we see, you know, similar results basically. Likewise, there are a couple of units where they have four different strains stocked. We still have the Chambers again, Nebraska. We've added Arley in yellow, Skamania in blue, and again, really not much difference. So the conclusions for steelhead is that all states are depending on one another's steelhead. We don't have really good survival in Green Bay. We have somewhat poor survival in this area from Algoma to Manitowoc, and really good survival in Indiana, southern Michigan waters, and the Wisconsin small trips. No differences among strain yet, though we might get there with more data for the Skamania trends versus Michigan winter strain. And our next steps, as I said, we're continuing to recover these fish through 2028. And we're going to start looking at strain-specific spawning patterns and seasonality of the catch rates. So what I want to conclude with, assuming I have time, Mark. All right. So this was something I added the last time I gave this talk, which was down to a steelheaders club in Algoma about a month ago. You know, the club manager, you know, I asked him, what are some questions your anglers want to learn about the fishery? So this is what they asked, and I thought it was interesting enough to share. So one of the things they were interested in is when will fish spawn out and die? And we have data on this for the 3 species that we tag. So for Chinook salmon, and a lot of people are surprised to learn this, is that most of those fish are spawned and done before they turn age 4. So those Chinook salmon go from very little to very big in just a few short years because they're basically lean, mean eating machines. What we see here are age in years and the number observed. Red are immature, black are mature. So we can kind of see at age 1, most of these fish are immature. At age 2, it's about 50-50. At age 3, they're mostly mature and ready to spawn. If for some reason they didn't spawn and they make it to age 4, they're pretty much all mature. But you can see here that most of those fish spawn out before they're age 4. Now, Chinook salmon only spawn once and die from the stress. Steelhead are different in regards to that. They can actually spawn multiple times in their life. And as a result of that, we do see a longer lifespan for these. So far, we've seen our oldest fish was a coated wire tagged fish that was 10 years old. And I mentioned it's coated wire tag because if it has a tag in it, we know when it was stocked. So we are 100% confident in that age. We do wild aging, but there is potential for error there with a coated wire tag, less so. And so what we see here, as I said, we do most of our analyses for recoveries ages 2 through 4, because that's when we see the most fish. But we do see a lot of recoveries up through age 7. And then after that, we don't see very much, but there's a handful out there. Interestingly enough, those mature steelhead, you know, we see, it doesn't, not until about age 6 do we see all the fish being mature. But ages 3, 4, and 5, we do see a substantial amount of mature fish. I will say some of this could be the time of year. So our techs assess maturity visually by examining the eggs. And if it's earlier in the year, it's really hard to tell sometimes if that's going to be a mature fish or not. But in terms of the age, you're looking at it. Now, lake trout are the longest lived fish in the lake. They're not. Sturgeon live longer. They're the longest lived fish of the 3 I'm talking about. And so the oldest fish we've seen from Lake Michigan is a 39-year-old. That was again from a coated wire tag. But they can live much longer than that. There's actually my boss, Chuck Bronte, he's got a picture. He's, I think, 55 or so at the time, holding a lake trout from Great Bear Lake, and it aged out to be the same age he was in the photo. So, you know, they can get up pretty far up there. But you can see in Lake Michigan, most of our catch, we're seeing 10 years or earlier. But this long tail is really important because these fish that are 8, 9, 10 years and older are the spawners. And these fish that are 15 and older are the ones that are devoting almost all their energy to reproduction. So those are going to be the critical spawners for producing wild fish in the lake. And you can see that bearing out in the maturity data. Black again is mature, green is immature. And by age 8, we're mostly mature. By age 10 and up, it's pretty much all mature fish. And they can spawn every year. Once they get to be a certain size though, they don't grow as much because they're doing most of its energy towards reproduction. And we're going to see that in this next step, because the other question I was asked is, can I tell a fish's age from its length? And, you know, generally there is a relationship between how old you are and how big you are. I mean, that's true for all animals. But with fish, it's more complicated for some species than others. So for Chinook salmon, because they grow so fast, their ages are pretty reasonable to estimate from their length. I can tell you most of the fish that are 10 inches or less are age 0, meaning they were
stocked or hatched that year. Most fish from 11 to 22 inches are age 1. Most 23 to 31 are age 2. And most fish 32 and larger are age 3. Now, I'm saying most because there is some variability here to note. So for example, at some of these marginal lengths, if we look at fish that are 31 to 32 inches, 55% of those were 2 years old, 43% were 3 years old. So yeah, most of them were age 2. But there were a fair number of 3-year-olds, and actually a small number were actually 4 years old. So get used to that uncertainty, because that gets more and more prevalent as we move on to steelhead and eventually lake trout. So one thing we learned right away with steelhead, well, not right away, but eventually enough, is that the different strains actually appear to have different growth rates. The first year we looked at results summaries, we only had age 2 fish, and we didn't see a ton of Wisconsin fish, and that was kind of a big deal. But Wisconsin stocks a lot of Ganaraska fish, and the Ganaraskas here in gray, and those fish at age 2 are only about 18 to 20 inches or so, which is right at the margin of where we start to really see them in the catch. And so by the time we got to the next year, we started to see more of those Wisconsin fish. I think part of that is because these Ganaraska fish are really small at age 2. You can see that Ganaraska does catch up, but you know, your differences here, Ganaraska was about 18 inches versus Skamania in blue was at about 23 inches. That's a pretty wide size range. So we have that variability in terms of looking at length at age for steelhead. And when strain is unknown, so if you're on the lake and you catch a steelhead, there's no way for you to tell what strain it is. If it has a tag, yes, you could pull out the coated wire tag, you could send it to us, you know, give me a call, maybe I could look that up. It's not something we generally do, but in theory, that would be the only way you can figure out the strain. And so if we remove that knowledge, for example, steelhead that are 29 to 30 inches, we have a few age 2s, 1%, 19% were age 3, 38% were age 4, 25% were age 5, 13% were age 6, and 4% were 7 and older. So you have 6 different age groups, 2, 3, 4, 5, 6, 6 different age groups within that 1 inch, 29 to 30 inch band. So it's really hard to tell a fish's age. But with steelhead, generally speaking, again, we can look at majorities. Steelhead that were less than 15 inches were usually a year old. And 16 to 23 inches are mostly age 2. Once we start getting older, get more mature fish, they're growing slower in terms of their length. So steelhead 24 to 26 inches were 2 to 4 years old. That's where we get that variability. And then 27 to 30 inches were mostly age 3 to 4. Everything over 31, 4 to 6. So I can give you, you can see I'm not being able to say, oh, this length is this age. It's more of an age range. And then lake trout, that length of age varies even more, as body growth size is slow in general, and pretty much nothing after the fish reaches maturity. It's just generating eggs. And so, for example, lake trout that are 29 to 30 inches in our data set ranged in age from age 4 to age 30. So, you know, and granted, those are some extremes, but that's a really wide margin. So generally speaking, I tried my best to be generally speaking, and at the younger end, we can have a better sense of this. So lake trout that were 16 to 19 inches are mostly 3 to 5 years. So again, Chinook salmon don't even live that long, and they're getting to be twice that size. So lake trout, yeah, they're not eating as much. They're slower growing based on their metabolism and their energetic needs. Fish, lake trout that were 20 to 25 inches are mostly 4 to 7 years old. And then I stopped trying to do this at 26 and larger. All bets were off. So our oldest observation at 26 inches was a 20-year-old fish. That age was known from a coated wire tag. So 26 inches isn't that big of a fish, but it could be up to 20 years old if you're a lake trout. So with that variability, really you need to look at a calcified structure or a tag or something that tells you the age, because the length really isn't going to be good enough. So with that, I'd like to thank you all for your attention. I'll stick around as long as you all want to discuss and answer questions.
Now I know why we hired him. It's an awful lot to keep track of. He does a great job. Are there questions?
They're doing this research in the Great Lakes, or doing fish migrate over here from, like, say, Lake Superior?
Are there any other lakes that they're doing this type of research on, and do any of those fish migrate over here?
Yeah, so that's a very good question. So there have been coated wire tags used in some of the other lakes for some periods of time.
but nothing like this mass marketing where we're tagging all of the fish that are in the stocked in a given lake at a given time. But that said, they had tagged Chinook and Lake Ontario for a set of year classes, and we did find, you know, a handful of Chinook salmon tags from Lake Ontario in Lake Michigan. So that does suggest, you know, that occasional fish is going to go on a, you know, a long sojourn into the unknown and end up wherever. Lake Superior, there are mostly lake trout stock there. We don't see lake trout move between the lakes very much because they don't move within their lakes that much either. We have seen the occasional Superior fish, but it's like one or two. And for Lake Huron and Lake Michigan, lake trout, it's like maybe a half dozen a year going between lakes among our observations. So, you know, a half dozen out of say 1,000 observations. So
what about salmon up there in Lake Superior?
Yeah, so salmon in Lake Superior, there actually aren't a ton of Chinook salmon stocked in Lake Superior anymore. In fact, there may not be any stocked in Lake Superior anymore, but they used to stock some. And I do know there are, you know, some Chinook and coho and spawning runs kind of up in the Wisconsin area, Lake Superior. I don't know how well those fish move out of Lake Superior. I mean, that's kind of, you know, my knowledge there is, you know, you're looking at the, you know, the Bayfield Peninsula. It's pretty west in the lake. Or Duluth Superior, which is as west as you can go. I don't know if those fish end up in Lake Michigan or not.
Where is the water temperature colder up there?
Lake Superior? Yes.
So that affects the production of the fish and what type of fish out there. I would think the salmon would like that cold weather.
Yeah, it's actually even colder than you typically would see for salmon. I mean, it's really good for the lake trout and lake whitefish, and they've always done really well. In Lake Superior, having snorkeled in both lakes, I can tell you Superior is substantially colder. One thing we notice if we look at invasive species, though, those cold water temperatures in Lake Superior, as a result of that, we don't see nearly as much aquatic invasives in that lake as we do in the lower 4 Great Lakes. And part of that's the cold water temperatures, and part of that is there's not as many populated cities where there's shipping. A good question, thank you.
So I heard a good segue there. You talked about invasive species. When the alewife populations drop, have you seen any evidence that the trout or steelhead or salmon can shift to other food sources like the zebra mussels or the quagga mussels?
All right, that's a good question, Carl. So yes and no. So we don't see the salmon and trout switch over to zebra or quagga mussels, like at all. And part of that's because these fish are largely piscivorous. They feed on prey fish. And with Chinook and steelhead and coho in particular, they are designed to look up, you know, forward and up, and their mouths are pointed in a certain way in their orientation. So they are feeding in the water column. So when they do switch prey, like steelhead, if there's not a lot of alewife, we do see evidence that they start feeding on a lot of bugs, particularly in Lake Huron. I think in the lowest alewife year, we saw, you know, 50, 60, 70% of the diets were invertebrates. And a lot of those are terrestrial, coming from the land, blowing off into the water, and then sitting there on the surface. You know, in some parts of the lake, it's gotten that high. Chinook salmon and to a lesser extent, coho, they still feed on alewife. Usually, I can tell you with Chinook, if we're not seeing a ton of alewife, those stomachs typically are empty, or they will have spiny water fleas, which is essentially empty. Spiny water flea is an invasive zooplankton, very small, and, you know, not very nutritious. The brown trout and the native lake trout, they are more generalist foragers. So they can feed in the water column, but they also feed on the bottom. And so it's not mussels, but they switch over to round gobies. We see, especially in the times of year when gobies are available. So round gobies exhibit an annual migration. So in the summer, June, July, August, they're near shore, shallow waters, spawning, and they're not being eaten by trout at that time because the trout aren't there. But in the winter, the warmest waters in the lake are deeper. And so in the fall, they migrate out deep. In the spring, they migrate back. And if we look at the spring diets, in some areas of Lake Michigan, lake trout diets are like 60, 70% gobies in April. So there is a potential, shift there based on forage availability. And I think what we see there is depends on the animal. Lake trout, was brown trout, steelhead, they're a little more generalist. The coho and the chinook are really looking for open water prey, which is mostly alewife, potentially smelt. Occasionally we see bloater, but the bloater tend to be deeper than the chinook are. So really it's alewife.
Any other questions?
What influence, what were the number of fish that were stocked?
What influences the number of fish that are stocked, right? Okay, so the stocking decisions, those are made by the states. State departments of natural resources determine how many fish are stocked. Now, Nick, I'm happy to pass the microphone your way if you'd like, but you can correct me if I'm wrong. Basically, in Lake Michigan, all 4 states and the tribal nations have representatives that sit on a committee, the Lake Michigan Committee, and they discuss and coordinate strategies for stocking that are based in large part on managing for predator-prey balance, which is affected by our estimates. Basically, we're looking at the estimates of the amount of biomass for Chinook salmon and comparing that to the estimates of the amount of biomass for alewife. A ratio of about 20 alewife per salmon is really good. A ratio of about 10 alewife per salmon is pretty bad in terms of the food supply being sufficient to support the fish. And so the target is somewhere between 15 and 20 alewife per salmon. So that information is taken in by the committee and used to make a determination on stocking levels. So again, those decisions were made at that state level and committee level. In the early years when we had, pretty low predator-prey ratio, like we had too many predators, there were some stocking reductions for Chinook salmon in 2013 and 2016. More recently, there were stocking increases, I think in 2019 and again in 2021. And that was because we saw the expected recovery in the biomass of the prey fish, and the decision was made that more fish could be supported.
Thank you.
Yeah.
So if there's anybody on Zoom who would like to ask a question, you can unmute yourself and ask it. Are there other questions here, Gary? I'll be right there.
What sense do you feel a Chinook salmon uses to move from Lake Huron to Lake Michigan to think it's going to get more heat?
That's a good question. That's a very good question. And I don't know. I know that Chinook salmon, they're known for having really good senses of smell. That is the sense that is used to pick up on the cues for natal homing. The technical term is olfaction, which is basically smell. I don't know if it's exactly the same as how we smell, but anyway, that perception is how they figure out where to go back to spawn. So maybe that's sensitive enough to pick up kind of signals of food abundance. Alternatively, maybe the fish stocked in northern Lake Huron move randomly and we just see the ones that come to Lake Michigan because they don't starve. I mean, that's possible too. We got to remember we're looking at our observations. And, I think it's likely, this is northern Lake Huron. They're pretty close to the straits there anyway. And I'm guessing that they pick up some signal and come into Lake Michigan, but I don't know that for a fact.
So, for fishermen, that are out there, do you ask them to send back the tags at all, or you just collect them when you're doing the surveys?
That's another good question. So there's a couple different programs. So Fish and Wildlife Service, we have our technicians out there at these cleaning stations. If we're interacting with an angler and we're allowed to sample their catch, we will collect the tags. We have a detector, so in the field, it's a strong magnet, can detect the presence of that metal tag. And if it's got one, we'll take off the snout and take that back to the lab. These are all fish that are being harvested anyway for consumption. So we're not killing any fish that aren't already dead. And basically we usually get the fish carcass from the angler after they've taken their fillets to collect our samples from. But there's also voluntary drop-off freezers, and the states organize those. You'll see signs at different cleaning ports and perhaps a chest freezer there. So if you are out there and you catch a fish, and you notice it has that adipose fin clipped,
it likely has a tag in it, and you can choose to cut off the snout, bag it, and put it in one of those freezers. It'll be picked up at some point, end up in our lab at some point, and coded as a voluntary angler return. So, you know, for those fish, we have some location data from the freezer where they came from. We can look at those tags for, you know, movement and stuff, but we can't include them for survival analyses because we don't know the effort that went into collecting them.
One more question. So why do you think evolution would drive the salmon, especially the Chinook, to die after spawning? It seems that, you know, so much effort goes into growing, you know, and then to die afterwards.
Yeah, it's different life history strategies. So for Chinook salmon, I mean, in the native Pacific, you know, they smolt out into the ocean. They spend a couple years just eating in the ocean. And then a lot of the spawning locations are very far upstream. So it's not necessarily that they want to die after spawning. It's not necessarily even evolution. It's just exhausting, you know, for a fish to swim, in some cases, you know, tens of miles upstream, maybe even more in the Pacific, you know, 10s, 20s, 30s, you know, I don't know the exact distance for sure, but, you know, it's a lot. And that takes a toll on these fish. Chinook salmon, their life history strategy is to get mature quickly. So that requires a lot of energetic costs metabolically. You know, like I said, those 3-year-olds are getting massive and they're coming up and spawning. And typically animals that grow fast don't live as long. So you know, the flip side of that is the lake trout. They're slow growing and they live a long time. So it's just different animal strategies, you know, like human beings and mayflies, right? Mayflies will be around for a year. We are around for much longer if we're lucky. So.
Any other questions? Here, let me get her first.
This may be very basic, but why Chinook salmon if it's not native? I'm new to this area too, so I'm not native either, but.
Yeah, all right. That's a great question. And it's a really cool story that I could spend a half hour talking about. But I will give you the Cliff Notes version. So the native lake trout, this is the apex predator, native lake trout in Lake Michigan. Lake Michigan actually supported the world's largest commercial fishery for those lake trout back in the day. But in the 1940s and 50s, we saw populations plummet, and there were 2 reasons for that. We had sea lamprey. These are these eel-like fish with the vampire mouths, and they were able to invade the upper Great Lakes when the Erie and Welland canals opened. So that circumvented Niagara Falls and allowed those lamprey, which were more or less native to Lake Ontario, or at least they always had access to Lake Ontario, to access the upper Great Lakes. And those lamprey attached on the lake trout, fed on lake trout, a lot of lake trout died. On top of that, we had fishing levels that were not sustainable. And one of the interesting reasons for that is that fishing production for lake trout increased in the Great Lakes in the 1940s because there was fear about German U-boats in the Atlantic at the time because of World War II. And that coincided with these lamprey. So we saw fishing go up, we saw predation of lamprey go up, and suddenly the bottom just came out. So by the time we get to the 1960s, we don't have very many lake trout at all left. Congress established federal hatcheries to raise lake trout. We started treating for sea lamprey to try to reduce those populations and work towards rehabilitating lake trout. But in the meantime, we had this other non-native species, alewife. I've mentioned them a lot. They became overabundant because we didn't have those lake trout around to eat them. So if you look at historical pictures from this area in the 60s, you'll see beaches that are just coated with alewife. I still remember that from when I was a kid. We still had a little bit of that, but in the 60s, it was really bad. And at Michigan DNR, Dr. Howard Tanner was the fish chief at the time, and basically he had the idea, kind of like a cattle rancher. Well, look at all this food, look at all this grass, we should have some cattle. Look at all this alewife, we should have a fish to eat them. We lost our lake trout commercial fishery, let's stock something
to try to establish a sport fishery, because this was hurt to the economies around here as well. You know, a lot of these local ports were based on the commercial lake trout fishery that wasn't there anymore. At the time, they were actually discussing striped bass, or I think it was either coho salmon or steelhead. I think coho salmon was the first one they introduced. They settled on Pacific salmon, introduced coho, then steelhead, then chinook, and that just took off. Lots of food for them to eat. Developed this sport fishery that at this point in time brings in $7 billion a year, Great Lakes wide. And so that's the Cliff Notes version of why Pacific Sea.
And at its peak, alewives has represented 85% of the biomass, fish biomass in Lake Michigan. So that's how massive they were. They're dying on the beach, stinking up and all that. So,
The Washington Island Sportsman's Club is very concerned about the number of cormorants. They're deforesting, devegetating the islands up there. And they're also concerned about the predation levels on fish. Are these birds, maybe I have to ask an ornithologist, but are these birds specifically feeding on one species of fish or all of them?
So I will preface this by saying I haven't studied cormorant diets in some time, but in graduate school, I worked on brown gobies, which are a little invasive fish. And as part of my research then, by just reading papers, I did read 1 or 2 cormorant diet studies, and they're pretty general, like yellow perch, alewife, gobies. Those were the main things that they were eating in this particular study. So basically, if you're small and you're a fish and you encounter a cormorant, you're screwed. Is kind of the summary of that. But you know, and cormorants, I mean, they're kind of a success story. Like I remember when I was a kid, that it was rare to see a cormorant, and you know they were protected, and now they're everywhere. So you could call it a success, but we hear this question a lot, and I know there's concern about abundance of cormorants. It's not my focal area, so that's about all I know.
So another round of applause. President Trout. I also want to acknowledge the team that puts together Fish Tales Lecture Series. Kyle Marina sitting here is right there. Crossroads, that includes Samantha Coyne, the CEO, and Lynn Mickelson, the administrative person in the office. And on the controls of the Zoom right now is Laura Kayakon from the library. And we also get help from Maggie Beam as well. So it's a group of us that get this done to bring this to you both in person and on Zoom. And we thank you very much for coming. And I know you asked a question how you can access the other videos. So actually, if you would just go to YouTube, that website, and search for Fish Tales Lecture Series, the county has developed a playlist, and you can just say Fish Tales Lecture Series playlist, and it'll pop right up, and all 24 or 30, how I don't know, all the way back when we started are there, and you can watch any of them, and you'll be able to see this one in a week or two when Laura has time to convert it to YouTube, and then we load it up there. It's a great partnership. We enjoyed very much. We enjoyed bringing this stuff to you, and thank you so much for coming and being so faithful.
Thank you, Mark.
Yeah.
I was trying to pull it up in live time, but not so much. Fish Tales Lecture Series.
Yep.
2 words, 1 word.
Just like that. And then playlist.
So there you go.
Yeah, there we go.
You put playlist after?
Perfect.
No, what happened? Well, sometimes you have to add Door County Library in there.
Yeah. Thank you all.